Okay, listen carefully. I got a extra credit opportunity for you, okay? If you will draw out on an 8.5 by 11 or 8.5 by 15, 16 piece of paper showing the process of DNA replication and labeling the parts, I will give you mega extra credit if you color it and then snap a picture of it and send it to my email. This is a reward for listening to this video. So the next thing we're going to talk about is how is the accuracy of DNA replication maintained? Well, the biggest way is using complementary base pairing. You know, the fact that A is always going to go with T, C is always going to go with G. Then another thing that happens is the DNA polymerases do a great job proofreading. If there are initial pairing errors, and there are, like 1 in 105 nucleotides, those are proofread and corrected. In the completed DNA, there's only one error for every 10 billion nucleotides. Mismatch repair of DNA will occur. Repair enzymes are going to correct errors in base pairing that happen to be missed by the DNA polymerases doing the proofreading. DNA can be damaged by exposure to harmful chemical or physical agents like X-rays, UV rays, cigarette smoke, and it can be spontaneously monitored and repaired by each cell, which is kind of interesting, and that's for your entire life. One way is called nucleotide excision repair. An enzyme called a nuclease will cut out and then replace the damaged stretches of DNA. Have a look at figure 1619, okay? When DNA nucleotides get altered, we call them a mutation. Mutation is when the DNA sequence changes become permanent and they get passed on to the next generation. Most of these mutations have no effect at all or are harmful. Very few of them are actually beneficial to the organism. But a very important part is, in spite of that fact, over time, these mutations are the ultimate source of genetic variation and lead to natural selection and ultimately evolution. Now we need to talk about an issue, that revolves around replicating the ends of the DNA molecules. This is a limitation of DNA polymerase, and it is not a problem for circular chromosomes of bacteria. It's a problem for linear eukaryotic chromosomes. The linear chromosomes have issues with completing the 5' ends. And after repeated rounds of replication, you get shorter and shorter DNA molecules with uneven ends. They're called staggered. Look at figure 1620. Telomeres are at the ends of eukaryotic chromosomal DNA molecules. They don't possess any genes, and they're typically just a multiple repetition of a short nucleotide sequence. In humans, it is typically TTA-GGG, repeated 100 to 1,000 times. Telomeres are important because they prevent staggered ends from activating the system that would completely stop the cell cycle or would put the cell into automatic cell death, apoptosis, genetically programmed cell death. They are a buffer zone that protects against shortening of the genes themselves. Now, please remember, telomeres do not prevent shortening of the DNA molecules. They postpone the erosion of genes near the ends. And there is some research that indicates this might be connected to aging. If the chromosomes of germ cells, remember, those are the ones that will be creating the gametes. became shorter in every cell cycle, then essential genes would eventually not be found in gametes. Telomerase is a very important enzyme that causes the lengthening of telomeres in germ cells. Shortening of telomeres might actually protect our cells from cancer because it limits the number of cell divisions. There is evidence, and this is very interesting, of telomerase activity in cancer cells that may allow the cancer cells to persist. And we're thinking that this presents a possible target for cancer therapy. So that's something to look for in the future. Remember, a chromosome is DNA plus proteins. Bacterial is double-stranded. It's circular with a small amount of protein. The DNA is coiled and then supercoiled in a region called the nucleoid. And remember, there is no nuclear membrane. Eukaryotic chromosomes are made up of linear DNA with a large amount of protein. DNA with its histone proteins added is known as chromatin. And I'd like for you to look at figure 1622. The unfolded chromatin is packaged into what is called a nucleosome. The nucleosomes actually look like beads on a string. Go ahead and turn to figure 16.22 and have a look at that. They are made of 2 each of the 4 basic histone types. And the DNA is wrapped twice around this core of 8 histones. The end tails of the histones protrude from nucleosomes, and we think that they are involved in the regulation of gene expression. Still have a lot of research to do in that area. Chromatin does undergo changes in how it is packed during the cell cycle. In interphase, some is organized into a 10 nanometer fiber. A lot of it is compacted into a 30 nanometer fiber that has folds and loops. Chromosomes occupy specific restricted regions in the nucleus so that the fibers of different chromosomes don't get entangled. It is most loosely packed in the nucleus. Centromeres and telomeres are highly condensed. This is called euchromatin. It is difficult for the cell to express the genetic information from these regions. I am so sorry, I just messed that up. The loosely packed that is in the nucleus is euchromatin, okay? Centromeres and telomeres are called heterochromatin, and those are the ones where it's difficult to express the genetic information that's in those regions. Sorry about that, guys. Histones can undergo chemical modifications, and this will result in changes in how the chromatin will condense, and this can also be an important source of gene expression.